Hello and welcome to the next in our series of Conversations with Giants in Medicine. I'm Ushma Neal from the Journal of Clinical Investigation. I'm happy to be joined today by Dr. Greg Semenza of the Johns Hopkins Medicine. It's a truth universally acknowledged that we need air to breathe, function, and thrive. But how do we sense oxygen and respond to a lack of it? Greg Semenza has made this question his life's work, and for the discovery of the hypoxia-inducible factor, HIF, he's being awarded tomorrow with the Lasker Award for Basic Medical Research. Thank you so much for joining me today. I really appreciate the chance to talk to you while you're here in New York.
Thanks for having me.
Can you tell me a little bit about your upbringing and what you were like as a kid?
Sure. I grew up in Tarrytown, New York, which is on the banks of the Hudson River, and went to Washington Irving Junior High School and Sleepy Hollow High School. I grew up with two brothers and two sisters. And my mom was an elementary school teacher, and my dad worked as a social worker at a hospital in New York City.
So what kind of an environment was that? Was it an academic one? With 5 kids, I imagine it was a survival kind of a mode.
Yeah, it was a chaotic environment. So, basically it was in the morning, get out of the house, and we'll see you at dinner time. So we had pretty much free reign in terms of getting around on our own, entertaining ourselves.
Were you a good student, motivated?
Well, it's funny because I sort of had this inflection point when I was in the 3rd grade, and we were supposed to read a book and then give a sort of an oral report on the book. And so it was my turn, and you would go up to the teacher, and she would ask you questions about the book. And I hadn't read the book, so that made it very difficult for me to answer the questions. And it didn't take her very long to figure out that I hadn't read the book. And she was very kind, and she said, well, maybe it'd be better if you go and read the book, and then we can talk about it after that. And so as a result of that, I became really a voracious reader, and have remained a voracious reader ever since then.
Oh, wow. So then when did an interest in science come along?
That was high school. So I had this fantastic biology teacher in freshman biology in high school, Rose Nelson, who is just this unbelievably talented, beautiful person who was able to transmit the beauty of science and the thrill of scientific discovery. And she had a PhD and had worked at Woods Hole, so she knew what research was. And that really was my inspiration. And so I had her as a freshman for biology, and then as a senior for AP biology. And so as a freshman, it was biology that I was hooked on, and then in my senior year, it was genetics within biology that I got really excited about.
So where in this trajectory did you decide that maybe you wanted to be a medical doctor?
Oh no, that wasn't until my junior year in college. So I was interested in genetics. I thought I would go to graduate school and study genetics. And then while I was an undergrad, a family that we were friends with had a child born with Down syndrome. And so that made me interested in medical genetics. And as a result of that, I decided that I would try to get into an MD PhD program and study genetics and also take care of people with genetic disorders.
Why an MD PhD? Why add the PhD on top?
Oh, because I was very much interested in research, and that didn't really change. So it was more adding on to that, the aspect of patient care. But sort of the PhD, the research part, that was always really my central interest.
Had you been exposed to someone that was, other than Dr. Nelson from high school, who was a scientist, and so you had a bit of a model to follow?
Well, again, through her help, I was able to enroll in a NSF-sponsored summer program at the Boyce Thompson Institute for Plant Research, which at that time was in Yonkers, New York. It was before it moved to Cornell. And so this course was held 5 days a week, Monday through Friday for several months in the summer. I'd take the train down to Yonkers, and we would do do experiments, and it was a lot of fun. It was really my first exposure to research and to experiments, and I found it very difficult, but I also found it to be really exciting.
We're jazzed by that.
Yeah.
So what did you choose to do the PhD part of your MD PhD focused on?
So the PhD story was a rocky road. My first lab that I went into, and again, it was in the context of an MD PhD, so you have all these concerns about getting everything done within a certain amount of time. That just kind of adds to it. So I was in a lab the first year, and a thesis lab. And this was a lab where I was like the first graduate student in the lab, maybe not a really good choice. and the project that I had was something that was very ambitious, maybe not a good choice, and things didn't work out. And I just got to the point where I felt that I didn't see this really working out, and maybe it was a good thing just to kind of admit defeat and move on. And I went and I spoke to my advisor, and he said it sounded like a reasonable plan. So I went to a lab at Children's Hospital in Philadelphia that was studying beta thalassemia, which at the time was sort of the first disease where the mutations that caused the disease were being identified, and the mutations being in the beta globin gene. And they had studied a number of families and identified the mutations that caused beta thalassemia in the families. And I was going to study this unusual family where one of the alleles was called the silent carrier, because normally you can tell from the looking at the blood cells whether someone is a carrier. And in this case, the father, who was an obligate carrier because he had 2 affected children, you couldn't tell he was a carrier. So it suggested that the mutation was in some way different. And this was going to be my project. And so the idea was you take the blood, isolate the DNA, make a library, pull out the beta globin gene, and sequence it. So I did all that, and at the end of 1 year, I got the sequence, and this was sequencing 4 kilobases of DNA, which at that time, that took you a year. That was a thesis, right? And so we got the sequence, and the mutation was the exact same mutation as the previous study, which meant that somehow the clone had become contaminated and the whole thing had been for nothing. Okay, so now we had 2 years, there's one lab and now a second year where I was going to have to start over for the third time. So I felt very depressed. And I had two students in my med school class. They were twins, and their father was on the faculty in the Department of Psychiatry. So I went to see him, and I told him what had happened, and he said, "Well, it sounds like you've got a good reason to be depressed." And then I didn't feel so bad. So I went back, started over again. Things went smoothly because, technically I'd already done it, and, we went through the project and got an answer.
So you finished your PhD, you finished medical school, and you end up for residency at Duke, and you decide on pediatrics. Why?
Well, because of course genetic disorders, many of them manifest at birth, and again with the Down syndrome being present at birth. So pediatrics seemed to me the most relevant area to specialize in for my clinical training, and I like kids, and so pediatrics it was a great experience to do internship and residency in peds, yeah.
And then you transitioned to Hopkins, and you've never left. So
no, I've never left.
So how did your interest evolve from thalassemia to hypoxia? Yeah,
so when I went to Hopkins, I had a PhD, and I was in a pediatric genetics fellowship. And I was thinking about what I was interested in, what would I like to study, which was a kind of odd thing, right, for a trainee to do, because it's usually, whose lab do you want to go into? So I was interested in the regulation of gene expression. I was interested in developmental regulation, And then from a sort of a technical standpoint, I was interested in transgenic mice as a way to study these things. And so that was sort of my interest. I was attracted to Hopkins because Haig Kazazian and Stelios Antonarakis, were kind of the leaders in the study of the molecular basis of beta thalassemia, collaborating with Stuart Orkin at Harvard to identify the mutations and then their functional significance. So that was one of the draws for me to come to Hopkins. And but by the time I got there, they had shifted from studying beta thalassemia and mutations in the beta globin gene to hemophilia and mutations in the factor VIII gene. And so, not being very creative, I said, well, I'll study the regulation of the factor VIII gene in transgenic mice. And so I called up the scientist that they had gotten their factor VIII clones from, and that his name was Chuck Shoemaker. At the time, he was at Genetics Institute in Cambridge. And I asked him, would it be possible to get these genomic clones to do this experiment? And he said, yeah, but there's this other gene you might want to consider called EPO. And he had also isolated the erythropoietin gene, but then, he's in a company, and they said, oh, now you have to work on, factor VIII. And so I went and I did reading about erythropoietin. I talked to one of the experts at Hopkins, Jerry Spivak, about it, and it seemed very interesting. And the part that, again, I focused on initially was the fact that it was expressed in the fetus in the liver, and then in the adult it shifted to the kidney. So I thought it would be very interesting to try to identify the sequences in the gene that were responsible for expression in the different tissues. And so that's what we did. We put different fragments of human DNA spanning the EPO gene into mice, and then looked to see if the gene was expressed and where it was expressed.
And somehow you stumbled onto the hypoxia responsive element in the 3' region.
Well, so of course the really interesting thing about the gene was that it was regulated by oxygen. And so that took me a few years to figure out. And so after we had done the work in the transgenic mice, identified the sequences that were necessary for expression in the kidney and the liver, then I wanted to try to understand the regulation by oxygen.
And at this point, you're still a postdoc?
Yes.
Or a fellow?
Yep, postdoctoral fellow, yep.
Okay.
So we had to try to figure out where the key sequences are, and of course normally people will look in the promoter region at the 5' end of the gene. But I did an experiment because I was very interested, again in terms of the regulation of gene expression. Was at that time the sort of the effect of chromatin was just being appreciated, and the technique of using DNA nuclease sensitivity assays to identify important control regions of DNA was being exploited to identify regulatory elements. And so I did this experiment with nuclei that I isolated from the liver of one of the transgenic mice. And there was a very strong DNase hypersensitive site within the human EPO transgene, but rather than being at the 5' end of the gene, it was actually in the 3' flanking sequence of the gene, which was kind of unusual. But I mean, that's what the data said. So we focused on that area and examined it in more detail and showed that we could take a piece of DNA that kind of encompassed that site and put it into a reporter plasmid, which at that time was a plasmid that encoded a protein called chloramphenicol acetyltransferase. It was an enzyme that acetylated chloramphenicol, and this was your assay, so there were no nice luciferase assays at that time. And we could show we could put it into the sequence into this plasmid, put the plasmid into a human cell line where EPO expression was induced by hypoxia, and now the reporter gene would be induced by hypoxia. And then we simply, made smaller and smaller fragments to identify the minimal sequence that was required, and we ended up getting down to 50 base pairs or 33 base pairs. And then we just started to mutate the individual nucleotides and found nucleotides that if we mutated, we completely lost the effect. And so we suspected that there was something a very important factor that was binding to that sequence that was responsible for turning on the gene. And so we tried to find that protein that was the key factor for turning on the gene.
So at this point, you've now transitioned to being a young faculty member and enter onto the scene your first great postdoc, Guang Wang.
Yeah,
right?
Guang Wang was my first postdoc, and one of the best postdocs I've ever had. He was just fantastic. Every experiment that he did from a technical point of view worked and was beautiful. And so by that time I had started trying to perform assays, binding assays, to look for a nuclear protein that would bind to this sequence. This was a real trial and error process. Guang took that up when he came into the lab.
So I love this story about how he's feeling very frustrated and you've got a stack of blots on your desk that he thinks are all negative results. But you found one where he's varying the salt concentrations on everything, trying to improve the assay and actually find this protein that's binding the HRE.
Yeah, so the problem is that the binding of proteins to DNA is very idiosyncratic. Lots of nuclear proteins are positively charged and they'll bind to negatively charged DNA, so you have to worry about nonspecific binding. But if you compete that off too much, you won't see the specific binding. So you have to kind of try to figure out what conditions will allow binding. And then we were looking for something that we hoped would be present in the nuclei from the hypoxic cells and absent from the cells that were not hypoxic. But of course that was a hypothesis, and that need not have been true. And so the idea was just to see some kind of faint signal that you could then try to optimize, because if you have no result, then you change the conditions. You don't know if you're getting closer or further away from where you want to be. But if you have some small signal, then you have something to work with. And he would do several of these experiments a day, and had this stack of autorads on his desk, and I was just thumbing through them one day, kind of holding them up to the fluorescent lights in the lab, and I came across this one gel where there seemed to be a faint band in the hypoxic lane, and I kind of held this up and I got all excited and I said, "Did you see this? Did you see this?" And well, of course, it was in this stack of failed experiments. So, and maybe it was more just hope on my part than anything else. But it turned out that was a signal that he was then able to optimize in a very short period of time to result in these really strong, definitive results of a binding activity that we called hypoxia-inducible factor 1.
Have you been chasing that moment again ever since then, that sort of aha, like, I think we found it? I mean, that must have been such an amazing moment to say, I think there's something there and I think we can find it.
Well, yeah, I was very fortunate because it was sort of at the beginning of my career, and you have a lot of both enthusiasm and confidence, and maybe haven't been privy to all the ways that things can not work out. But it was. But the way science works, of course, is that there are all these small steps, and then you don't really kind of appreciate the process until you're looking back at an outcome from some distance.
So maybe the aha moment was a little delayed because it HIF turned out to be a dimer, right? And it was incredibly difficult to figure out.
Well, so, the gel shift gave us an activity, but of course we wanted to actually isolate, physically a protein and the sequences that coded for the protein. And I was very influenced by Steve McKnight, who was at the Carnegie at that time and had developed a technique for expressing proteins in bacteriophage, and then having that expressed protein detected by a radioactive DNA probe as a way to identify transcription factors. And so we tried to use this approach, and we made a library from hypoxic cells, and we screened with the probe because we had done an experiment that we thought indicated that we could do, that there was a single protein that would be recognized by the probe. And we screened millions and millions of clones, and we got no positive result. And so, again, at some point you have to decide, do we keep trying, or do we stop, admit defeat, let somebody else do it, or maybe try some other alternative strategy, experimental strategy, which of course was biochemical purification. And at that time, we were kind of a molecular genetics lab. We didn't even own a fraction collector, so really, nothing in terms of doing biochemistry. But fortunately, Tom Kelly's lab was right across the street, and his lab had been one of the first labs to purify a protein based on its DNA binding activity. And so his lab was really helpful in doing that. And we ended up purifying the protein from 100 liters of HeLa cells in suspension culture. And we were able to purify enough of the protein by this DNA affinity chromatography to get some sequence data. And we had done experiments that told us that the protein did not stay on DNA very long. So it went on, it came off very rapidly, which is the worst thing you could possibly have if you're doing an affinity purification, because we knew as we were pouring the stuff in the top of the column, it was coming off the column, So, but we were able to get enough protein to get some DNA sequence data and to isolate the cDNA.
Okay, so is that finally your moment for champagne?
Well, so to get the cDNAs, that was a big moment because it enabled us to generate antibodies, to look at RNA, to look at protein, and also kind of share those reagents with lots of other people so that they could do the same. And so that really opened the field once we had those reagents. Yeah.
So do you ever wonder how it might have been different if you didn't call it HIF and instead you called it something else like Jigglepuff or something? Because I imagine you must say HIF more than any other word.
Yeah, well, I stand by it. People said, well, now we know it's induced by growth factors. We should call it something else. Well, I think its physiological role is to be induced by hypoxia. So I'm happy with the name. And then the other thing was, people said, when I named it HIF1, they go, oh, is there a HIF2? Where's HIF2? Of course, there wasn't a HIF2 at that point, but now there is. And there's also a HIF3. So it turned out to be a good name.
All right. So having a new lab at this point, you're still a really young PI. I mean, yes, you had your failures during your PhD, but you're on a roll. Then shortly thereafter, you also created the knockout such that you could see that HIF was interacting with VEGF.
Yeah, so that was one of the first experiments that we wanted to do to get a sense of what was the importance of the factor, and we had already been doing experiments that told us that that the factor didn't just turn on EPO, that it had many other functions. But the knockout mice, that's sort of the way that you really find out about the importance of the factor. And by that time I had hired my third postdoc. So my first postdoc, Guang Wang, was involved in the purification My second postdoc, Binghua, was involved in the isolation of the cDNAs, and then my third postdoc, Narayan, was involved in the generation of the knockout mice. We also collaborated with John Gearhart's lab, as we had with the transgenic mice, on generating the knockout mice. And so those knockout mice turned out to, the mice that were homozygous for the knockout allele were embryonic lethal and had defects both in the heart, the production of blood, and the blood vessels. So all three components of the circulatory system were dependent on HIF1 for their normal function. So that was a really beautiful finding because we already had, of course, the involvement in physiology, and then this told us about involvement in development, and really sort of showed that it's all just a continuum.
Yeah.
Right.
So is there anything that HIF doesn't do? So in preparation for this interview, I read a lot of the things you wrote. And so here, I know maybe I'll show the figure online, but there's this figure from Nature Reviews Cancer showing HIF in the middle and every single gene and different part of, cellular function that HIF is involved in. Is there anything that HIF doesn't touch?
Well, then you have to ask, is there anything where oxygen is not playing some sort of a role? And the answer is oxygen is so fundamental to life that there's tremendous crosstalk, and so HIF really is a nexus for many different inputs and outputs in order to what we think of the classical homeostasis, physiological homeostasis. Yeah.
So where do we stand with using HIF as a therapeutic target?
Right. So now you say, well, but it does many things. You may want to target it for one purpose, but can you do that without having other effects that will be deleterious? But I think the answer is it's coming along. So for example, it's really satisfying because we started with EPO. And understanding the regulation of the EPO gene, and then now it looks like drugs that will stimulate HIF activity will be used as agents to stimulate erythropoiesis. So EPO is a recombinant protein. It requires injections on a several weekly basis, and the idea is you could simply take a pill that would stimulate HIF activity and would stimulate erythropoiesis. And that seems to be, that's in clinical trials now, so I think those drugs will probably come on the scene within the next few years. And then they may also be useful, for example, in treating ischemic conditions, although I think that'll probably take longer to show that whether or not there's benefit there. And then the flip side, of course, is cancer, where the cancer co-opts all these normal physiological responses for its own purposes, and where the idea is that inhibiting HIF activity may be very useful for treating cancer, because most of the standard chemotherapies target dividing cells, which are the well-oxygenated cells in the tumor, and the hypoxic cells are resistant to those therapies.
A different question. You chose to do the MD and the PhD, and you were clinically active for many years, but are no longer. Do you think people need both anymore?
Well, again, it's a question of what are your goals? So my goals were always to try to bridge the gap between the basic science and the clinical. And so for me, it just made sense to have both an MD and a PhD. To be in both worlds, to try to make that bridge that gap, and that's remained for me the most satisfying thing. So we've done, it's been the beauty of the project is that we started out with very fundamental molecular questions, and we can follow that all the way to the clinical, and for me that's been really fantastic to be able to do that. And so for what I wanted to accomplish, I think it was the best career path. But there are many people, their career path, may not really make sense. And to be honest with you, I don't really think it makes sense to get the MD if you don't want to take care of patients. I think there are enough graduate programs where you'll learn the basic science. Certainly at Hopkins we have a program in molecular medicine where you can do that without getting an MD. So I think, there's no point in going to medical school if you don't want to take care of patients.
So I know you've probably done this entire body of work without thinking about winning prizes, but you've won the Wiley Prize, you've won the ASCI Prize, the Gardner, and tomorrow you'll be collecting the Lasker Prize for Basic Medical Research Award. What do these kinds of recognitions mean for you?
Well, I think it's really great for people to get the sense of research and the path of research and how very fundamental discoveries can, lead to very practical results ultimately. For the public to get some sense of what science is like. And when you can make that connection to the diseases that, most people end up being affected by, heart disease, cancer, the public can really appreciate that. This is why it's important to support science. And in terms of our own field, I think we, yes, there were all these awards, but we kind of flew under the radar for quite a while. And, we were making really dramatic progress. I'm talking about the whole field now. And still kind of flying under the radar. And now I think, obviously, that's over. The field will get, there'll be even more interest in the field, and it'll just continue this exponential growth, that's been going on for the last 20 years, really.
No, it must be really gratifying.
Oh yeah, absolutely. Because we started with a very focused question. It's not like we're asking this big question, but it was a very focused, simple question. And that's the way science works. It's the serendipity of it that makes it so exciting, because you never know, where things are going to lead. And you think but most of the time, at least for me, it turns out no, your hypothesis is wrong, but it doesn't matter. You found out something really interesting. That's what makes science so exciting, because you just never know where it's going to lead.
So if you had to do it all over again, you couldn't be an MD or a PhD scientist. What else do you think could have kept you this passionate as a career?
Yeah, well, I guess the one thing I pretended when I was younger, I guess, was that I was an FBI agent, and so investigating crimes, being a detective. It's like being a scientist, right? So that would probably be it. Yeah.
Fun. And do you have a joke you'd be willing to tell on camera?
Oh, sure. Yeah, I have a joke. My daughter told me this joke many years ago. And it was the guy's driving down the road, and he sees a farmer and a three-legged pig. And he said to the farmer, "What happened to the pig?" And the farmer says, "That pig saved my life. I was out tilling the crops. My tractor fell over. I was trapped on top of it. That pig pulled me out and saved my life." The guy says, "Well, that's very interesting, but what happened to the pig?" The farmer says, "That pig saved my life. I was sleeping, the house caught on fire, the pig woke me up and dragged me out of that burning house. That pig saved my life." The guy says, okay, but what happened to the pig? What happened to his leg?" The farmer says, a pig that good, you eat him one leg at a time."
All right. It's been a delight having you here today and getting a chance to hear a little bit more about your life. So thank you so much, and congratulations again on the Lasker Award.
You're welcome. Thank you.
Thank you.